I want to say, it's the first time I'm here at this conference, and it's a great pleasure and a great honor to actually be here and to share some of the work that we're doing. I was quite surprised first that I was invited because this is kind of like an ocular conference. I'm not really working that much on lutein. I'm mostly working on the provitamin A carotenoids and retinoids. But nevertheless, there's kind of like some commonality kind of like in terms of the bioavailability when it comes to carotenoids that I kind of like trying to bring across today. So in terms of the kind of the talk today is we're kind of going to look at how we can actually best measure bioavailability by using kind of traditional but as well kind of like new and upcoming technologies that will enable us to do this kind of like much more eloquently and quicker, and also making it much more accessibility, or making it much more accessible in terms of field trials, which kind of like is really where I'm trying to develop new technologies to actually make it much more accessible in the field. So in terms of the kind of like the topics I'm going to talk about today is first of all kind of talking about bioavailability of lutein and beta-carotene. Then also kind of looking at the current methods and their limitations, the use of stable isotopes and why I think this is a really neat way of trying to address this question. And then also trying to show you that actually we can overcome the problems in field settings by measuring bioavailability and bioconversion by just simply using single blood measurements and getting the exact same information that we get by taking a multiple range of blood samples and area under the curve. So I'm kind of like showing that to you in the end. So first of all, what I think I will be talking about, but also Thorsten, who's going to come after me, is going to talk about the same way, is actually the question of what is bioavailability. If you look into the literature, and if you even ask colleagues, and you kind of look into what happens in pharmacology and what happens in nutrition, there's not really that much of an agreement. And if we kind of just look at the definition here, it's the rate and the extent to which an active drug or ingredient from a drug or a modality from a drug is absorbed and then becomes available at the site of the action. And in our case here, it would be, for example, the amount of lutein that's absorbed and actually then becomes available in the brain or in the ocular tissue. So that's kind of like really the question. But then what we also need to do is we kind of need to think about the whole process. And the process starts already in the gut itself. We talk about bioaccessibility, which is actually how much of the components that we have in our diet actually will become available to actually cross the barrier into the intestinal cell, or kind of like how are assessed from the food itself. So the bioaccessibility is important. And then the crossing into the intestinal cells, which actually already is what we call the bioavailability of these compounds. Then depending on the compounds you're looking at, you actually have either just lutein that actually just kind of gets across into the plasma, or when it comes to the provitamin A carotenoids, they actually need to be converted as well to a certain extent. And we see this happening anything between 50% to 90% of the absorbed carotene actually is converted. And then from the plasma, they need to be secreted, or it's not secreted, but taken up by different tissues. And then we have re-secretion of certain components back into the circulation. And all of that together makes what we call the bioefficacy that actually was defined by van Lieshout already kind of like some years ago. But it's actually a combination of bioaccessibility, bioavailability, and bioconversion. So it's kind of like a complex process. Now the question is, how do we measure that? Kind of like in a human situation. Now, in most cases, what we actually do is we measure the area under the curve, as you can see here. Some beautiful examples of where you can like use isotope research-based methods, where you're kind of looking at whole blood isotope contributions, but also kind of like chylomicron-rich fractions. And I'm going to come back to that in a minute, and to kind of like to show the benefits and the weaknesses of these kind of like methods. What, however, I think is really important to understand is that when we're talking about these carotenoids, is that they are so lipid soluble that actually very often we forget that once you have these carotenes in your diet, they actually kind of like arrive in your enterous site and quite often will sit there, because depending on the chylomicron capacity to deliver them to the bloodstream, you quite often have what we call a second meal effect, i.e. you just simply have a little bit of fat that actually then pushes these carotenes out. And in cases where you give very high doses, in this case here 120 milligrams, you actually see that the area under the curve after the second meal, which didn't contain any carotene whatsoever, is even larger. So that kind of just indicates to you that the impact of diet, kind of with and without the compound, is really, really important. And what drives me mad is that when you look at literature, that quite often people don't even describe the amount of food that people had and at what time point. And that makes it very incredibly difficult to see whether that increase in the second meal effect is actually driven by the meal or it's actually driven by the compound itself. So we don't actually really help the public in understanding what really happens physiologically when we don't properly describe the process as actually what should be happening or what is happening. The other important thing also that actually this brings across is that actually because of this effect that we actually see when you have non-labeled compound that you have on a day-by-day basis or day-by-day diet, you need to ideally have what we call a washout period. And as you can see here, you want to kind of avoid carryover effects that you actually have from anything that was given before. Now, that could be in your habitual diet, but also that's really critical. If you can like do a crossover experiment in your volunteers, you must make sure that whatever you've done before is not carrying over into your next experiment that then actually then blurs your data in terms of what you're actually going to get out of it. And therefore, for example, when it comes to lutein, we are talking about a 14-day washout period that's been recommended by Thurman et al. Because what we actually know is that the half-life of lutein is about 5 days, and about by 14 days, 90% of the lutein would have been eliminated. So that's kind of like, it's not everything is gone, but the majority of it is gone. So that's kind of like a relatively safe period. And even if, kind of like in some trials, we kind of like see that that is taken quite carefully off, sometimes the weeks become a little bit short even then to actually make sure that this actually is properly done. So the next question that we need to then kind of like do in terms of looking at the methods that we have is what kind of bioavailability assessment methods do we have? So first of all, what we do is we use the area under the curve concentration, but this can be different approaches. The most used approach is what we call the partial AUC, or the AUC that's run for the day when you actually run the experiment, up to 8 hours, 10 hours, 12 hours, 6 hours. You see actually a massive range in the publications that you see, and it's very often driven by practicality, i.e. how long can I actually use my nurses? How long is my center open? How long will my volunteers be prepared to stay in the unit? That are kind of very important practical questions that sometimes even drive the way we design our experiments. But then on the other hand, you can get these beautiful experiments where people actually have looked at, what happens in terms of when do I get my half-life? When do I get my Vmax of my compound of interest? And therefore, I can actually determine what is the actual time point that I need to have before I reach saturation within my plasma sample. As you can see here, that can take quite some time. And for most carotenoids, that we're talking about 6 to 8 weeks before you actually get to that level of absolute saturation. And therefore, when we can look at the recommendations when it comes to area under the curve, we can do the short or partial AUC, but in fact, actually, what is recommended is that the partial AUC should be truncated at the Tmax, i.e. the time to achieve maximum concentration, and at least we need to have 2 quantifiable samples that need to be collected before we expect the Cmax, the maximum concentration, therefore to estimate the partial AUC. So these are kind of like recommendations that are made in terms of the way we should analyze things. Now, the question is, how is it actually done? I've kind of just summarized here 4 examples as to how we see these bioavailability studies being done on lutein in the literature. So we see here, for example, a partial AUC, 2 different examples. This one here is a partial AUC where, in fact, lutein was just given around 3 milligrams, but people were put on a lutein-deficient diet for about 2 weeks, so lutein concentrations dropped, and then they were put on the 3 milligrams, and the serum was just simply measured as is. So we're just kind of looking at total concentration of lutein over a period of time and then get the area under the curve. In this case here, which is a bit more eloquent, is what actually has been done. As you can see here, we're looking at lutein in the chylomicron response. We're looking at newly absorbed lutein over a period of time. But again, kind of like it's relatively short here. Therefore, it's a partial AUC. Other studies actually kind of have looked at the long-term effects, as you can see here nicely. But the problem with this study here is that we kind of have a long-term AUC, kind of not really AUC, because we only really have a beginning and an end point, i.e. we can't really calculate the area under the curve, and we therefore don't really know whether or not we've actually reached saturation at this time point. Whereby in here, because of the variety of different time points, we can actually know, yes, we got to the Cmax, and we can actually calculate adequately the AUC. So these are the different things that you actually find. We've also done similar studies with beta-carotene. As you can see here, we've looked at 3 different formulations that were tested for DSM, and we also looked at how long it actually takes for actually reaching saturation. As you can see here, very, very similar to lutein. We actually needed at least around 50 to 56 days before it actually happened. But at the same time, and I come back to that later, in my other experiments that I'm going to show you, is that even with stable isotopes, we've now got to the conclusion that stable isotope work ideally need to be followed up for a longer period of time. The earlier studies were done for a relatively short period of time, but we now understand, particularly when it comes to retinoid metabolism, we need to be able to be in what we call the terminal slope, to be really able to understand the kinetics of the components that actually we're looking at. The other critical thing that we need to think about when it comes to bioavailability is that very often, not always, but very often, the compound of interest actually is in lower concentration when it's absorbed than what's already in the serum. So if you really want to see how much has been absorbed, you're going to have to use the chylomicron fraction, because otherwise you're not going to see what's been absorbed at the time when you actually gave the dose, if that's the question of interest. And even then, we need to be aware that there are limitations. Because if you look at the literature of most separation methods that are used, the chylomicron separation methods are what we call triglyceride-rich lipoprotein fractions. And the separation methods do not allow us to separate between the chylomicron remnants and the VLDL, which are slowly coming up from the liver as you go along. And that is very, very critical, because as you can see nicely in this diagram, we are interested in what is absorbed, but what happens as you go along in the timing, from around 6-7 hours onwards, on general, you actually start seeing that absorbed chylomicron particles, in our cases now lutein, beta-carotene, are resecreted by the liver based on VLDL and are unseparable in our fractions. And therefore, we are actually diluting the effect of absorption, and therefore overestimating the amount of the compound that has been absorbed, because we are mixing the VLDL with the chylomicron fraction. And therefore, our area under the curve are much higher than they actually should be. And I can show you actually later on how we potentially could overcome this problem. Now, what we think very strongly is that you can make life a lot easier if you can use stable isotopes. Why? Because I don't need to use chylomicron separations. Because once I give an isotope, I can separate the isotope just simply based on mass, and I can follow that up over a period of time, and I know exactly what's been absorbed and know exactly what's in the system. And here's just an example where you see, for example, beta-carotene here nice in green. I see also here retinol esters and retinol coming up that actually have been absorbed in a human. And not only that, but what you can nicely see, if, for example, I'm interested in the bioconversion rate of beta-carotene, I see here retinol as the control substance here, the 10 times labeled retinol, beta-carotene given and being cleaved to form 5-time retinol, as you can see in this diagram here. So if you have beta-carotene 10 times labeled, it's cleaved right here. It produces a 5-time labeled retinol, and I can actually follow this up beautifully by looking at the retinol being formed from my beta-carotene. Because the masses are significantly different, I can detect exactly how much retinol was produced from beta-carotene, how much retinol was absorbed. And if I look at the ratio of these two over the period of time, and you can see here we've done this in the first study for 14 days, I can see very nicely that this person here is very well bioconverting and absorbing the beta-carotene, whereby this person is not. So just simply looking physically at the responses that you see, you can actually see, yes, my goodness, there's a clear indication. But not only that, what we've realized actually is that the curves run completely parallel. And we came to the idea, why do we not just simply take a specific time point in which we measure the ratio between these two components and then come up and see whether that agrees with the area under the curve that we've measured over time, and therefore could potentially reduce the effect. And in fact, it's true. If you can't measure the bioefficacy by using the AUC method and you compare that to just simply a bioefficacy method that just uses one time point, we see perfect agreement between the two methods. So that kind of is an indication that this potentially works. In terms of the bioefficacy method, what we also wanted to do is actually to kind of like see, can we simplify the area under the curve method for bioefficacy? And what we've actually done here is to kind of like look again very similarly the traditional AUC method. So you're kind of like looking at the area under the curve for your retinol esters plus kind of like over the area under the curve of retinol esters plus beta-carotene, i.e. the products of beta-carotene, and to see how much actually has been converted. And if you do that at a particular time point, you can actually then like look at whether or not you need to calculate the area under the curve or whether you just simply can actually look at one particular time point. And if we can like start doing that, we actually can like see very nicely here that if you can like looking at the conversion efficiency rate, it starts very, very high and then starts dropping, and you can see here it starts dropping very, very fast, i.e. the conversion rate goes down. If you kind of look at the area under the curve, we also see it's very high at the beginning. Of course, kind of like everything is similar at the beginning, and then it starts dropping, but not as quickly as if you're using just simply one time point. Now, the reason why this drops not as fast as this is because you don't really see the effect of one single time point because you're looking at the area under the curve, and the discrepancy towards the end of the curve actually is much, much clearer if you have a single time point than if you actually have an area under the curve over a period of time. So that kind of like makes sense. But what I want to show you here, and that's critical, is that an area under the ratio at time point 6 hours after you've given the dose is non-significantly different as if I take the area under the curve of 8 hours or at 6 hours. And that kind of like quite often is used in research. So you can like we see a lot of papers that actually look at area under the curve of 8 hours, and we can now show you that you could get exactly the same result if you just simply take one blood sample, take the isotope ratio, and you get exactly the same result for that. The next question we were asking ourselves is that the problem that you see here is that the beta-carotene concentration keeps on creeping up. And we know that from this time point onwards, that actually we don't really look at chylomicron responses anymore. This is a combination of chylomicron plus VLDL. And the question is, how can we overcome this? And you see here, what we've actually done is we've just simply assumed, and you can see actually in our next slide, that this is not just an assumption, but is based on real observation. You see here that if you kind of look at the retinol ester concentration and the peak of it, in terms of when actually these compounds peak, you see here the beta-carotene and the retinol esters peak always at the same time. So they behave like in the earlier phases where you're only looking at chylomicron fractions, i.e. the ratio stays the same. The ratio, however, then diverts later up because your retinol esters are absorbed by the liver but not secreted out on VLDL, they're secreted out on RBP. And therefore, I don't have that dispersion anymore between the retinol coming in and getting out of the liver. So it's a clearer picture. Whereby with beta-carotene, you don't see that. You have a mixture between chylomicrons and VLDL, and they completely mix, and you don't see the difference. So if we just make the assumption that the ratio should always stay the same because they're both bound on chylomicrons, and we then calculate this out, we then come to this situation where we can actually calculate out that beta-carotene, with the help of the retinol ester concentration, actually behaves like we see retinol ester behaving, and we then can also calculate out the amount of beta-carotene that's now bound on VLDL. Now, if we do that, and we come back to the calculations, we see here actually that the adjusted area under the curve, surprise, surprise, actually gets to about 78 or 77% and completely stabilizes out. That means if I can calculate out and correct the carotene that's truly been absorbed, I see actually that my conversion efficiency is stable throughout the period of when I'm actually absorbing the beta-carotene. That makes perfect sense. But not only that, we can actually see also that the actual ratio that we have at 6 hours perfectly agrees with what we see at 8, 10, and 12 hours in terms of our adjusted area under the curve calculations of bioconversion. So it kind of works really, really nice in terms of being able now to use a single factor to tell us something about the conversion efficiency that we see in these individuals. And kind of to conclude, what kind of we now have shown actually is that kind of depending on the different time frames that we actually have on this curve, we are now able to look at carotenoid conversion with one time point. We are able to look at total body stores of vitamin A with one time point. And we are also looking at the total ability to kind of look at bioefficacy, either combination of bioaccessibility, bioavailability, and bioconversion at a later time point. And the reason why you have to go later is because you have post-absorptive conversion that is still happening inside the body that we know kind of more or less stabilizes at around 14 days. So what I'm kind of have shown you, what I tried to show you today is, first of all, that the area under the plasma concentration time curve is used quite a lot, but that we should at least have 3 to 4 samples during the terminal log linear phase to be able to kind of look at whether or not we've reached saturation in the compounds of interest. Or at least to use something, if you have to have a short partial AUC, use something where you know what that curve is so that you actually have a control that you can compare your compounds to. For the estimation of AUC, we should have at least 2 quantifiable samples that should be collected before the expected Cmax. We also know that the intestinal bioconversion of beta-carotene can be estimated based on a single plasma sample, which will make life a lot easier in field settings, but more importantly, it will now allow us to look at what happens in children, because in children you can't take more than 2 blood samples at any time. So therefore, this now is an applicable analysis method that we can actually apply in field settings in lower income countries. And then finally, if you cannot afford or you, for one reason or another, can't use isotopes, our modified method is potentially a nice way forward to actually correct the area under the curve for carotenoids that otherwise slightly will overestimate the actual absorption rate in that sense. So I want to finalize by thanking all the collaborators, all the funders that have helped us in our research, and more importantly, I'd like to thank my Newcastle team that have done all the hard work in doing all the isotope analysis, but also all the modeling that was done in collaboration with Penn State University. Thank you.
Thank you very much. This paper is open for discussion.
Thank you very much. Very interesting presentation. I was wondering, can you buy this isotope-labeled compounds, or did you synthesize them, or?
No, they are commercially available. We don't make them. They are made by a company in the Netherlands called Buchem. And they actually had a team, or they made from a team that was based at Leiden University, who actually developed the synthesis rate for both carotenoids as well as retinoid. And in fact, actually, they can create any kind of carotenoid that you are interested in. So it's not, they not just do beta-carotene, they do a range of different compounds too.
Thank you very much. I think your paper has a lot of implications for everything we've been doing within this field, and this goes for everything across, large longitudinal clinical trials where we do one blood sample test, HPLC, separate carotenoids, area under curve report. And one of the things I've seen from day one is that the expected correlation between serum and tissue concentrations, in this case the macular pigment that we measure, it's pretty weak, to be honest. And I just wonder, is it simply down to how we've been measuring carotenoids in blood up to now, and does your work have major implications for that?
That's a really good question, but not one that is easy to answer. Because there are so many different factors, as you rightly say. For example, the first factor that comes to my mind is obesity or adiposity. And we know that carotenes or adiposity or fat cells work like a sink. And we need to take that into consideration. So that kind of is just one little factor that's critical to kind of look into. The other factor, as you rightly say, is kind of the question is if you once you start reaching an equilibrium, what does that actually mean in terms of the tissue concentration? Does that mean actually that whatever you see in plasma is actually then mirrored as to what you actually see in the tissue? That could be the case, but it could also not be the case. And we know from work that, for example, the group in Marseille has done that there are genetic factors that may kind of influence the way that relationship plays out in the long term. So there's a lot of work that needs to be done before we can be confident that just simply measuring what you see in plasma is what you should see in a tissue. And I think that because you made that connection, and I think the good news is that in terms of measuring the tissue, it's an absolute nightmare when it comes to humans, but you have the gold method because you can measure the concentration in the macula non-invasively. There's not a lot of work that can be done non-invasively, and that's definitely one of the areas that should be looked into. And where we should harmonize methods, which I think is not yet happening, and I think we absolutely must do that. Harmonize methods and then compare with real hardcore tissue measurements to see what is the relationship and what are the factors that influence that relationship to better understand the implications. Because to me, to just simply drive plasma levels up and then expect we're actually going to see health outcome is just a very simplistic way of looking at the implications.
Yeah, I agree. Thank you. Yeah,
and I'd like to add to that, I mean, because we know that lutein, for example, really circulates in HDL, and we saw with Christine's talk, how the lipoproteins are important for getting the lipid into the retinal tissue. So it's not just plasma, but how is it circulating in the plasma? Right.
Thank you very
Okay,
much.
thank you.
Thank you so much.